There is an animal you have seen thousands of times, in aquariums, in restaurants, in the ocean, in your own home maybe, in a glass bowl on a shelf. You have looked at it your entire life and assumed you understood it. You don't. Not even close. In the last 20 years, scientists have run study after study on this animal and come back with the same conclusion, over and over again. Everything we thought we knew about fish is wrong. They don't sleep the way we thought. They don't feel the way we thought. They don't remember the way we thought. They don't sense the world the way we thought. And they can die in a way that should be impossible for something that lives entirely underwater. 5 questions. 5 answers that will completely change the way you see the most common vertebrate on Earth. This is Abyss Files. Subscribe if you are new here. You will want to stay for all 5 of these. Let's start with the one that sounds the simplest. Do fish sleep? You have probably never thought about this question seriously. And if you have, you probably landed somewhere around, probably not. They don't have eyelids. They never stop moving. They live underwater. Sleep seems like a land animal thing. You are wrong. But here is the part that makes it interesting. The reason you are wrong tells us something profound about what sleep actually is, and whether our definition of it even makes sense. Let's start with what we think sleep is. When a human falls asleep, several things happen simultaneously. The eyelids close, muscle activity drops, heart rate and breathing slow. Brainwaves shift from the rapid, chaotic patterns of waking consciousness to slower, more synchronized rhythms. The body moves through cycles of light sleep, deep sleep, and REM, rapid eye movement sleep, where dreams occur. For most of the history of sleep science, this definition was the entire definition. Sleep was those things. All of those things together. Fish fail almost every single criterion. Fish do not have eyelids. They cannot close their eyes. Fish do not have a neocortex, the region of the mammalian brain most associated with the complex neural activity of sleep. The brainwave signatures that define human sleep stages simply cannot be measured in most fish because the relevant brain structures do not exist in the same form. For decades, this led scientists to conclude fish do not sleep. They rest, maybe, but not sleep. That conclusion is now known to be wrong. Here is what changed. In 2019, a research team at Stanford University published a study in the journal Nature examining sleep in zebrafish, a small freshwater fish that has become one of the most important research animals in neuroscience. Using microscopic imaging technology that allowed them to watch individual neurons firing in real time, the researchers documented something remarkable. Zebrafish experience 2 distinct sleep states. The first looks like slow-wave sleep in mammals. Reduced brain activity, decreased responsiveness to the environment, lowered heart rate, minimal movement. The fish float almost motionless, sometimes wedged into a crevice or resting on the bottom. Their metabolism drops. Their bodies slow down. The second state is something the researchers had not expected to find: brief bursts of neural activity during sleep, rapid micro-movements of the eyes, muscle twitches, body posture shifts. In other words, a state that looks, neurologically, remarkably similar to REM sleep, the sleep state where humans dream. Zebrafish appear to have a functional equivalent of REM sleep, and zebrafish are not unusual. They are representative. Evidence of sleep, true sleep, not just rest, has now been documented in every vertebrate species studied to date. Everyone. But here is where it gets truly strange. In March of 2024, researchers at the University of Basel published a finding that shook the sleep science community. They discovered that some fish do not need orexin to stay awake. Orexin is a neurochemical that regulates the sleep-wake cycle in mammals. In humans, the loss of orexin-producing neurons causes narcolepsy, the inability to stay awake. Orexin was considered so fundamental to vertebrate sleep regulation that scientists assumed all vertebrates must use it. Fish broke that assumption. Some fish regulate their sleep-wake cycle through a completely different mechanism. A mechanism that does not exist in mammals. A mechanism that, until 2024, nobody knew existed. What this means is not just that fish sleep. It means that sleep itself, one of the most universal behaviors in animal life, evolved multiple times through multiple different biological mechanisms, and we have been studying only one of them. The fish in your aquarium is sleeping right now, just not the way you think. Before we get to question 2, I want to plant something in your mind. In 1976, researchers published a paper suggesting that fish have a memory span of approximately 3 seconds. You have heard this. Everyone has heard this. It is one of the most widely repeated facts in popular culture. It is completely, demonstrably, thoroughly wrong. We will come back to this. And the truth, which scientists have known since the 1950s and 60s, but which somehow never made it into public consciousness, is going to make you question every other fact you have casually accepted about the animal world. But first, question 2. Do fish feel pain? This question has implications that go far beyond biology. Every year, between 1.2 and 2.3 trillion fish are killed by humans. That number is so large it is essentially incomprehensible. It is more than all the mammals, birds, and reptiles killed by humans combined, multiplied by orders of magnitude. And until very recently, the scientific consensus was, it does not matter. Fish do not feel pain. They do not have the neural architecture for it. A fish on a hook is not suffering. It is responding reflexively. There is nobody home. That consensus is wrong. Let's go back to the year 2002. Dr. Lynn Sneddon was a researcher at Liverpool University studying fish neuroanatomy, the physical structure of fish nervous systems. She was not looking for pain receptors. She was not trying to prove anything about fish suffering. She was just looking. What she found stopped her research cold. Fish have nociceptors. Nociceptors are the specialized nerve endings that, in humans and other mammals, detect potentially harmful stimuli. Intense heat, intense pressure, caustic chemicals, and trigger the neural cascade we experience as pain. Before 2002, the scientific literature contained no evidence that fish had these structures. It was assumed they did not. They do. Sneddon published her findings. The response from the scientific community was skeptical. Having the hardware for pain detection does not prove the experience of pain. Maybe fish have nociceptors, but do not consciously process the signals in a way that constitutes suffering. So she ran more experiments. She injected acetic acid, vinegar, into the lips of trout. The fish showed something she had not expected. They rocked back and forth on their pectoral fins. They rubbed their lips against the gravel and the sides of the tank. They stopped eating. They became hypervigilant, showing dramatically heightened responses to any stimulus in their environment. These are not reflex behaviors. Reflexes are immediate and automatic. What Sneddon observed was sustained, prolonged, complex behavioral change that persisted long after the initial stimulus. And then she administered morphine. The behaviors stopped. When she used naloxone, a drug that reverses the effects of opioids, the behaviors returned. The fish had pain receptors. Those receptors triggered neural signals. Those signals produced behavioral responses consistent with the experience of pain. Those responses were suppressed by painkillers and restored when the painkillers were blocked. This is the same mechanism at every step as pain in mammals. In 2019, Sneddon published a meta-analysis of all existing research. The conclusion was unambiguous. Fish possess the anatomical and neurological machinery for pain. They exhibit behavioral responses consistent with pain. Those responses are modified by painkillers in exactly the way you would expect if pain were genuinely being experienced. A study published in Scientific Reports found that when rainbow trout are slaughtered using conventional methods, they experience between 2 and 22 minutes of what the data suggests is severe pain before death. 2 to 22 minutes. For one of the most commonly consumed animals on the planet. And most people have never heard of Lynn Sneddon. There is a reason this research has not been widely publicized. There is a reason it does not appear in school textbooks. There is a reason the question of whether fish feel pain is still treated, in casual conversation, as obviously answered by no. The answer has been yes for over 20 years. Fish feel pain. And the scale of what that means, given that we kill between 1 and 2 trillion of them every year, is something our civilization has not yet begun to seriously reckon with. Now let's talk about memory, and about the most durable myth in popular science. Goldfish have a 3-second memory. You know this fact. You have always known this fact. It has been in cartoons, in television shows, in casual conversation, in 1,000 metaphors about forgetfulness, for as long as you can remember. It has no scientific basis whatsoever. None. Scientists have known this since the 1950s. The myth does not come from any study. It does not come from any observation. It comes from the casual assumption that a simple-looking animal in a simple environment must have a simple mind, and from our collective guilt about keeping those animals in tiny, featureless bowls. Here is what the research actually shows. In 2008, a team from Plymouth University trained goldfish to press a lever to receive food. The lever only delivered food during one specific hour of the day. Within weeks, the fish had learned when feeding time was. They began congregating near the lever before it activated. They left when the feeding window closed. They remembered the time of day, every day, for months. A study published in 2022 trained goldfish to use landmarks to navigate a tank. The fish could accurately gauge distances and judge positions based on visual cues. When the landmarks were changed, the fish adapted, using learned information in new situations rather than following a fixed pattern. They were not memorizing a route. They were building a spatial map. In another experiment, researchers played a specific sound when feeding goldfish. 5 months later, 5 months when the fish were released back into their natural environment and heard the same sound, they returned to their original feeding location. 5 months. Goldfish remember specific acoustic cues associated with food for at least 5 months. And goldfish are not exceptional among fish. They are below average in cognitive complexity compared to species like wrasse, which pass the mirror self-recognition test, a benchmark of cognitive sophistication that only humans, great apes, dolphins, and elephants reliably pass. Or cichlids, which recognize individual human faces. Or archerfish, which learn to identify and shoot precisely targeted jets of water at specific insects they have seen before. Here is the part of this story that nobody tells you. The goldfish memory myth spread because it was convenient. If goldfish have a 3-second memory, then keeping one in a small, featureless bowl is fine. The fish forgets the bowl is small before it has time to notice. No guilt, no welfare concern, just a cheap, easy pet. If goldfish actually have memories that span months, if they can get bored, if they can recognize patterns, if they notice when their environment is repetitive and understimulating, then putting one in a small bowl is something else entirely. The myth was not just wrong, it was useful, and we believed it without ever asking for evidence, because believing it was easier than the alternative. We are halfway through. 3 questions answered. 2 remain. If you have not subscribed to Abyss Files yet, do it now. Every video on this channel is built around the same idea. The things we think we know that turn out to be wrong. Question 4. How does a fish actually experience the world? You see through eyes, you hear through ears, you smell through a nose, you feel through skin. Fish have equivalents of all of these. They see, they hear, they smell, they feel. But fish have something else. Something you do not have. Something no land animal has. Something that perceives a dimension of the physical world that you are biologically incapable of directly experiencing. It is called the lateral line. Run your finger along the side of a fish. If you look closely at a goldfish or a trout, you will see a faint line running from behind the head to the base of the tail. A slight change in scale color, a barely visible seam. That line is an organ. It is a system of pressure-sensitive cells called neuromass, arranged in a channel that runs the length of the fish's body and extends onto the head and face. Each neuromass contains hair cells that respond to disturbances in the water. Not sound exactly, not touch exactly. Pressure changes in the water around the fish. Every object in the water displaces water as it moves. Every current, every eddy, every heartbeat in a nearby organism creates pressure waves that spread outward through the water. The lateral line detects all of it. A fish swimming in total darkness can detect the exact position, size, and movement direction of every object within a certain radius by reading the pressure map of the water around it. Not by looking at it, not by hearing it, by feeling the way the water is moving in 3 dimensions around it. This is a form of perception that has no human equivalent. We cannot imagine it because we have never experienced anything like it. And here is what the lateral line actually gives a fish in practice. A school of fish, 100, 1,000, 10,000 individuals, can turn as one body in milliseconds, changing directions simultaneously with a precision that would require split-second communication between every member if it were coordinated consciously. It is not coordinated consciously. Each fish is reading the pressure waves produced by the fish around it through its lateral line. When one fish turns, it produces a pressure change. Every fish within range detects that change and responds. The response itself produces pressure changes that cascade outward through the school. 1,000 fish turning as one body is not a decision, it is a wave. When you watch a school of fish change direction in the ocean and feel that particular sensation of wonder, you are watching pressure physics. You are watching the lateral line doing exactly what it was designed to do. But the lateral line is only one of the extraordinary sensory systems fish use to perceive their world. Some fish, including sharks, rays, and several freshwater species, have an additional system called the ampullae of Lorenzini. These are small, jelly-filled pores clustered around the head and snout that detect electrical fields. Every living organism generates a weak electrical field simply by being alive. The electrochemical activity of muscles and nerves produces electric potentials that radiate outward into the surrounding water. Sharks can detect these fields. The great white shark's electroreceptive system is sensitive enough to detect electrical fields of 5/100th of a billionth of 1 volt per centimeter. A human heartbeat from 5 meters away produces an electrical field roughly at that level. A shark can sense your heartbeat from 5 meters away, through walls, through sand, through complete darkness. A fish holding perfectly still in a dark crevice is not invisible to a shark. It is broadcasting its location continuously to every nearby electroreceptive predator. There is no hiding. You are being perceived right now by systems you cannot perceive. The ocean is full of them. And now, the last question, the one that sounds the most impossible. Can fish drown? Think about that for a moment. A fish, in water, drowning. It sounds like a joke. It sounds like the setup to a riddle. Of course a fish cannot drown. Fish live in water. Drowning is what happens to things that cannot survive in water. The answer is yes. Fish can drown. Not in the way you drown, but the outcome is identical. Oxygen deprivation. Inability to extract enough oxygen from the surrounding medium. Death by suffocation. Here is why it is possible. Fish do not breathe water. This is the key misunderstanding. Fish breathe oxygen. The same oxygen you breathe. The same molecular oxygen that your lungs extract from air. The difference is that fish extract it from water rather than air. Water contains dissolved oxygen. Oxygen molecules that have physically dissolved into the water from the atmosphere and from the photosynthesis of aquatic plants. Fish gills are extraordinarily efficient extraction systems, pulling dissolved oxygen out of the water and transferring it into the bloodstream. But dissolved oxygen is not guaranteed. It is not unlimited. It can run out. The amount of oxygen that water can hold is dramatically lower than the amount of oxygen in air. Air is approximately 21% oxygen by volume. Well-oxygenated surface seawater contains about 8 milligrams of dissolved oxygen per liter, 26 times less. Fish are operating on a fraction of the oxygen supply that land animals take for granted. And when conditions in the water drop that fraction further, fish begin to suffocate. Pollution reduces dissolved oxygen as bacteria consuming organic waste use up what is available. Algal blooms triggered by agricultural runoff consume oxygen as they decompose. Warm water holds less dissolved oxygen than cold water. Fish killed. Events where large numbers of fish die suddenly in a body of water are almost always caused by oxygen depletion. Not poison, not disease, drowning. Every summer, hypoxic dead zones form in the Gulf of Mexico, where the Mississippi River discharges nutrient-rich agricultural runoff into the sea. The dead zone in 2023 covered approximately 8,776 square miles. 8,776 square miles of ocean where fish cannot breathe, where they drown. And here is the detail that makes this stranger still. Fish can also drown in air. When a fish is removed from water, its gills, which require water to maintain their structure, which collapse without the support of water around them, stop working. The delicate tissue that maximizes surface area for gas exchange in water becomes a liability in air. The lamellae, the thousands of tiny folds that make gill breathing so efficient, stick together, collapse. The surface area for oxygen exchange drops catastrophically. The fish suffocates in the element we are adapted to breathe. It drowns in air. While we can drown in water, fish can drown in air. Two life forms, both absolutely dependent on oxygen, both capable of drowning, each in the element the other breathes. The same physics, the same outcome, the same terrible end, just in opposite directions. Let me bring this together. We started with 5 questions that seem simple, 5 things that everyone thinks they know about an animal they have seen their entire lives. Do fish sleep? Yes. Through mechanisms so different from a mammalian sleep that they force scientists to redefine what sleep fundamentally is. Do fish feel pain? Yes. They have had the necessary neurological hardware for pain detection for at least 500 million years. We have known this for over 20 years. We have chosen not to act on it. Do fish have a 3-second memory? No. They have memories that span months. In some documented cases, years. The myth exists because it was convenient, not because it was true. How do fish experience the world? Through sensory systems so alien to human experience that there is no adequate metaphor for them. The lateral line reads 3-dimensional pressure maps of moving water. The ampullae of Lorenzini detect electrical fields so faint that a human heartbeat is audible from 5 meters away. Can fish drown? Yes, both in water that has run out of oxygen and in air that their gills cannot process. 5 questions, 5 wrong answers that we have been carrying around as facts. Every wrong assumption we made about fish was based on the same error. We looked at an animal that did not look like us, did not behave like us, did not live where we live, and we assumed it was simpler than us. We were wrong. There are 35,000 known species of fish, more species of fish than all other vertebrates combined. They are the most numerous, most diverse, most widespread vertebrate on this planet. And we have spent most of human history looking at them through glass and assuming we understood them. We did not. We are only now beginning to. The animal in the bowl on your shelf is sleeping right now. It remembers the last time you fed it. It can feel the vibration of your footsteps through the water. It has been there the whole time. You just were not looking closely enough. Subscribe to Abyss Files. The ocean keeps showing us things we were not looking for. We will keep going back.